All right, here we go on part 2 of chapter 7. Let's get started. So here's where we left off. We've gone through glycolysis, pyruvate oxidation, citric acid cycle, and we're about to get into the electron transport chain. Now, I want to remind you at this point, from a single glucose, that's been completely broken down. We produced a little bit of ATP and carbon dioxide from those 6 carbons in glucose. There's 6 carbon dioxides. And where most of that energy is now at this point, again, a little bit of ATP, but most of the rest of it is tied up here in these electrons that have been ripped out of glucose and those other things that we've produced along the way. And those can now be fed into electron transport chains. So this, again, goes back to this idea. We're not losing that energy in an explosive release of heat and light like the burning wood, but we're going to squeeze a little bit out of those electrons to do work, right? That idea of using energy to do work. So let's look at the electron transport system where we're going to pass these electrons along and use that energy. All right, so we're in the mitochondrion. These proteins that pass these electrons along, they're embedded in the inner membrane of the mitochondrion. So that's what you're seeing here. There's the outer membrane, and here's the inner membrane, and here are those proteins. These proteins are called cytochromes. That's, you'll see, just so you know, you'll see those names and be aware that those are the kinds of proteins involved. They're electron carriers, but they're also proton pumps, as we'll see here in a second. As these electrons are passed along, these are all redox reactions, right? One compound is losing the electrons, something else is picking them up. And at every step, every time those are passed, think back to one of the questions I asked on the previous lecture, every time the more electronegative thing is picking up the electrons, some energy is lost or can be used in some way. Again, most of it's still lost, but some of it can be used. Ultimately, pass electrons down the chain. Ultimately, yoink, they're going to end up being picked up by oxygen. And remember, those protons go along with the electrons, so oxygen picks up the electrons and hydrogens, and it becomes water. But look here. Do you see ATP anywhere? No. We have not produced any ATP. We've passed these electrons along. They're off to water. Like, wait, what happened? Where's our ATP? So what these proteins do, or a few of these proteins do, they're proton pumps. So while this electron passing is happening, 3 of these will use that energy to move protons, into that intermembrane space. So the matrix here in the mitochondrion, it's sort of like the main cytoplasm of the mitochondrion. And then that intermembrane space, that's where it is more acidic. There's more protons, and that creates a gradient. And that is really ultimately the key. And so creating that gradient, we have a coupled transport system. You're like, wait, what's coupled transport? Go back and look at the chapter on membrane transport, active transport, and coupled transport. We've talked about this already. We've even talked about proton gradients being used to do things. This is called the proton motive force. So these protons can be used to do other things too, but they're really important in generating this ATP. So we've got this gradient. Gradient equals potential energy. What does that potential energy do? We see protons zipping back through this protein here. Guess what this guy is called? ATP synthase. What does it do? It makes ATP. It synthesizes ATP from ADP and phosphate. So this is that oxidative phosphorylation, not substrate level that we saw in glycolysis and the citric acid cycle. I encourage you to check out various links to animations that I've got on here. I think that can really help. Here's just a different cartoon. Again, maybe that helps you visualize it, but really take your time just to look at it. Nothing is appearing or disappearing out of nowhere. Just follow the electrons. NADH passes off its 2 electrons here. Those get fed along the chain. Those electrons go to lower and lower energy states. Where does that energy go? Some of it's lost as heat. but some of it's used to pump protons. Here we're actually seeing the intermembrane space is at the bottom here on this figure. And then that potential energy is now used to make ATP. So most protons come back down through this ATP synthase, and that is what drives this. And this is a protein that actually kind of, as the protons go through it, rotates and generates ATP. Again, check out some of those videos. That'll help your understanding. Okay, review time. In the complete reactions of aerobic respiration, all of what we saw, energy from most ATP produced is from what? All right, hopefully this was a slam dunk at this point because you've heard me say it many times. Most of the ATP produced is from that proton gradient. It's the gradients produced from the energy from those electrons, but ultimately it's the gradient that drives that formation of most of the ATP. We get a little bit of ATP from glycolysis and citric acid cycle. Most of it, though, comes from what we just talked about. All right, let me give you another question. As we kind of start to look back and account for what's going in, what's going out, and where things flow, let's first think about where the electrons go. So most energy flows in this sequence that was tied up in glucose. It's passed to electron carrier, electron transport chain, that proton motive force, proton gradient, used to make ATP. Wonderful. But where did the electrons go? So given your choices here, where did they go? All right, maybe you had to go back and look in your text at some of the figures or a little bit of trial and error on this question. But think about what's happening, right? Originally, those high energy electrons are in glucose. They're passed to an electron carrier. Some end up in FADH2, so that's another electron carrier, but most of them are with NADH. Those electrons get fed into electron transport, and again, they don't make the ATP directly. They end up binding with oxygen. Oxygen ends up picking them up to produce water. All right. So here's where we get all our ATP throughout this breakdown of a sugar. How much do we get? We get a little bit, again, citric acid cycle or Krebs cycle in glucose. And most of it comes from that last step, chemiosmosis, from those high energy electrons feeding into the electron transport chain. Of course, not all of the energy is used to generate ATP. Some of it is lost as heat. So only about 34% of that energy from that broken down glucose molecule ends up in ATP. So just like a little bit like the fire example, we're still losing energy as heat. This is not a perfectly efficient process. And how much can we get? In theory, you can get 38 ATP in a bacterium, 36 in eukaryotes, but the actual energy yield is a little bit less. And one of the big reasons for that is that inner mitochondrial membrane, where you have this proton gradient, it's not impermeable to those protons. So they can leak a little bit across the membrane, and that you sort of lose a little bit of the power of that gradient when that happens. Why the difference between bacteria and eukaryotes? In eukaryotes, you have to move that pyruvate from the cytoplasm into the mitochondrion, and so you lose a little bit of potential energy as a consequence of that. All right. So now, we've seen this idea of co-transport, that proton motive force can do other kinds of work, as we saw before. Here's a proton pump moving protons into this outside the cell in this example. And what is that potential energy being used for? In this case, it's being used to move something like sucrose into the cell. So again, we've focused on this as energy production, but that proton gradient can be used to do other things. Okay, given the list, what of the following can be used in cell respiration? Which compounds can be used? All right, so you might have initially just jumped on carbohydrates because that's what we've been talking about. But remember back many slides ago, I talked about how other compounds feed into cell respiration too. So any of these kinds of macromolecules can be used in cell respiration. It's just they may feed in at a different place in glycolysis or the citric acid cycle. And we can see that here. So here's our different macromolecules being broken down and feeding in at different places, ultimately producing carbon dioxide in a lot of cases, water, and ammonia. Now, if you're really looking for a good time, you can check out one of these interactive metabolic maps. And, I sort of show this to for almost shock factor. That all these pathways are interconnected, though. That's the idea. You see these arrows going around everywhere. It's like, oh my gosh, what's happening? But yeah, your cells are complex, and what happens in them, there's a lot of things that can be happening. But what to sort of just take from this at a glance is the connectivity. These arrows connect in a lot of different places. One step in one metabolic pathway, the product from one step, might be used in a completely other pathway. We look at it in a class like this in a very simplistic linear fashion, but these pathways are interconnected. The other thing to remember is this: highly regulated, these pathways are highly regulated. They're multi-step. We saw this idea of feedback inhibition in the last chapter. ATP, for example, it inhibits a step of glycolysis. And that makes sense, right? If you've got a lot of ATP, you don't need any more. So ATP acts as an inhibitor. You're like, plenty here, don't need any more. Whereas, say, AMP, adenosine monophosphate, it's a stimulator of this pathway, because it's like if you got a lot of monophosphate, that probably means you don't have a lot of ATP. And so that's going to stimulate this. Citrate. Got a lot of citrate. That means, hey, slow this whole thing down. We're producing too much citrate in this first step of the citric acid cycle. Slow the rest back down up here. So that's that negative feedback. So what happens when oxygen isn't present? And you've heard about these before in the previous video lecture. Fermentation and anaerobic respiration. That allows cells to continue to generate energy even when oxygen isn't present. And you need oxygen for this electron transport. So here's electron being passed, ultimately passed to oxygen. If there's no oxygen, that's going to make this come to a grinding halt, right? So the electrons will just stop being passed from one thing to another because they're all just backed up. So you need oxygen for electron transport to work. So without it, there needs to be other ways to generate energy. All right. But look at glycolysis. Is oxygen needed anywhere here? No. You could still do glycolysis without oxygen. And you generate a little ATP, you got to spend some, but you can generate some ATP too. But what do you need for glycolysis? So basically, what do cells require for glycolysis to work in the absence of oxygen? All right, so given the choices there, the correct answer is they need NAD+, that electron carrier, because you're trying to pull these electrons out of what you're breaking down, starting with glucose. If you don't have a place to pass them to, then you're stuck. And here's why you might not have NAD+. So when oxygen is present, electrons get passed, and this NADH can continue to feed its electrons in here, producing NAD+. But with no oxygen, this all backs up, and then NADH cannot pass off its electrons, and you do not get NAD+. So you're stuck with a bunch of NADH, reduced NADH, carrying its electrons. And again, you need NAD+. You don't need oxygen, but you need NAD+ for this to work. Here just a different way of looking at it here. NAD+ is necessary. So cells have a way of generating that NAD+ even when oxygen isn't present. So electron transport is shut down. That's not working. They can still take pyruvate, and when oxygen isn't present, they'll use pyruvate as an electron acceptor. We'll see how that works, as opposed to oxygen, because there's no oxygen in this case. All right, so here's one way living things can do this. So here we've got pyruvate, and step one here in this case, alcohol fermentation, carbon dioxide has to be taken off of pyruvate here. And notice it says 2 carbon dioxide, but this is also saying 2 pyruvate. Again, thinking if we're starting with 1 glucose, that's how many pyruvate we'd have. So that will produce acetaldehyde, and then that is what electrons get passed to. So here's NADH, 2 of them, again, 2 acetaldehyde, passing its electrons off. And when acetaldehyde is reduced, gaining electrons, that produces ethanol. All right, so that's why when you think of what yeast do when you're brewing beer, here's, carbonation, carbon dioxide, and ethanol as a consequence of fermentation. But more importantly, pay attention over here. By passing the electrons from NADH, we've now generated NAD+. Now glycolysis can work. It can take those electrons from glucose and pass them to this electron carrier. All right, so your cells don't do alcohol fermentation, but they have a way to get around this as well. This is lactic acid fermentation. It looks very similar. So here's glycolysis proceeding through those earlier steps, producing 2 pyruvate for each glucose. And pyruvate directly now is accepting electrons from NADH. So just pick 2, each pyruvate picks up 2 electrons. So 2 pyruvate, 2 times 2 is 4 electrons. And that produces lactate. So that's why you think of, this person, I'm showing a person running here, think of like lactic acid buildup in your muscles, as that's a response to anaerobic respiration. So the oxygen-starved tissues are still finding a way to make ATP. Again, the important part here is we've continued to make NAD+, and that can now feed into glycolysis because you need that. All right, here's a little more lateral thinking question. Why is glycolysis considered to be one of the first metabolic pathways to have evolved? So a few choices here. This is a little more, put a little more thought into this one. All right, so here were your choices in some order. We can go through some of these. Glycolysis, D, relies on chemiosmosis. No, it doesn't. So that we can rule that one out. Found in prokaryotes but not eukaryotes? No. I mean, we're talking about what's happening in a mitochondrion, or in the cytoplasm rather, of a eukaryotic cell. So that doesn't work. Let's see. B, it doesn't involve organelles or specialized structures, does not require oxygen, and is present in most organisms, right? And so that's the one you ultimately ended up with. And that's the correct answer. And this all makes sense. So early life, there was no or almost no oxygen in the atmosphere. The fact that most organisms do glycolysis suggests that this is something common to all living things. It doesn't require any organelles, so you find it in all kinds of living things. All right, let's talk about oxidation without O2. So we've talked about fermentation. That's where we saw organic molecules like pyruvate or acetaldehyde picking up electrons so we can regenerate NAD+. But other compounds can be sort of like oxygen, where we saw that electron transport chain. Other compounds could be at the end or somewhere else along that electron transport chain and act as a receptor. Now, that doesn't necessarily happen in our cells, but I'm talking about in all living things. We see actually quite a range of how organisms use carbon sources to generate energy and build ATP. So these would be the anaerobes. So a lot of prokaryotes, or there's many groups of prokaryotes where we see anaerobic cellular respiration. So the methanogens, for example, they use carbon dioxide and reduce it to methane. So that's why they're methanogens. They generate methane. because carbon dioxide is their electron acceptor. Sulfur bacteria, they use sulfate as an electron acceptor, and that produces H2S. So that's that sort of rotten egg smell, hydrogen sulfide. And so you can almost see a similarity here. It sort of looks like H2O a little bit. So same kind of thing. You're getting two protons. See, there's protons coming along with those electrons, just like we saw with oxygen picking up electrons and hydrogens, and we have H2O. So there's all kinds of pathways, mechanisms that have evolved in cell respiration. The first one on the list is, say, our example. We're organotrophs. We use sugars, and from that generate carbon dioxide. And the electron acceptor is oxygen, which picks up the electrons, and then we have H2O. Let's see, this is let's pick a different one here. Organic compounds. So using sugars and other things, just like we do, producing carbon dioxide. Okay, this is looking familiar, but here's the difference. Instead of oxygen, these guys, iron reducers, they're passing it off to these iron ions and producing here this reduced form. So there's all kinds of ways that organisms do this. Why is oxygen so great? Well, here's that iron example. Here's the free energy difference from glucose to iron. You pass those electrons off, and you have the potential to squeeze this much energy from that. Nitrate, a little better, right? There's a bigger free energy difference when you're moving those electrons from glucose to nitrate. But oxygen gets you even further. And so this is definitely an advantage from, think, natural selection, evolution perspective. If, when these kind of mechanisms evolved, it would have the possibility to outcompete these other methods because they can get so much more energy out of a single glucose than these other pathways. All right, so that's it for cell respiration. We'll see you next time when we talk about photosynthesis.